The core principle of PCR is the use of an enzyme called DNA polymerase to make a copy of DNA strand. In the first PCR step, the double strand melts to single-stranded DNA, a step known as denaturation. The DNA polymerase enzyme gets to work and copies the single strand molecule, starting at the bound primer region. In practice, two different primers are used: a forward primer that binds to one strand and a reverse primer that binds of the opposite strand. The choice of primers is important as each primer binds the DNA in a specific place. PCR is used in search of several deletions, mutations, and polyforms in a single gene. Simultaneous analysis of multiple molecular markers for molecular cloning, exploring colonies to detect the presence of grafts, saving time and money. For the realization of PCR is required DNA, the sample that will be amplified, magnesium chloride. It increases the temperature of DNA irradiation and the fundamental for the optimization of the reaction. Buffer PCR solution that resists pH change and maintaining the same pH in a reaction. Mix of DNTPAs, for example, DATP, DGTP, DCTP, and DTTP. Primer forward and reverse. It serves as the start of DNA synthesis. Tag polymerase enzyme that carries out the replication of the DNA sample. And water grade molecular biology, dilution of all regions. To achieve the design of primers, some software such as Snapgene and NCBA can be used, in which the gene to be amplified is introduced and the indicated primers are obtained. To explain the obtaining step by step, the following is done. 1. The coding sequence of the gene is obtained in FASTA format in NCBA. 2. It is copied and pasted into a work file. 3. For the first forward, the first 18 13 nucleotides in the direction are selected. The TM is calculated. 4. Seek possible secondary structures in oligoanalyzer. 5. For the first reverse, the same steps are carried out. 6. The vector to be cloned is selected. 7. To declone it into a vector, the restriction enzyme found in the vector is added and its sequence is bound to the first one. Each one has the corresponding restriction enzyme sequence It is indicated with a point where it is going to cut to enter the vector. 8. The vector sequence is searched and placed in Word. The position of the restriction enzymes that were selected using a gene is searched. 9. In Word, the position of the enzymes is identified. In the position identified where it cuts, the gene sequence is introduced with the design primers. 10. The entire sequence is entered into SnapGene to observe the simulation of the cloned vector. Finally, with the primers used in practice, a PCR can be performed to know the sex of a person from whom only one sample is taken.
Tau tempered DNA, 10x stock buffer solution and 50 millimolar magnesium chloride solution at room temperature and placed on ice. To prepare the master mix solution for PCR, calculations were performed to determine the proportions and concentration of each of the reagents which are shown in Table 1. Once the necessary quantities of reagents are placed, mix the master mix by inversion. Divide the master mix into 3 aliquots to obtain a positive control, a negative control, and that of the sample to be analyzed. Add 2 microliters of molecular biology water to the microtube that will contain the negative control. Must be molecular biology grade to avoid contamination and amplification of non-specific fragments. In the microtube of the sample to be analyzed, add 2 microliters of extracted DNA that is the template for the identification of the CFX or CFY gene sequence. In the microtube of the positive control, add 2 microliters of DNA solution containing only the amplified of 447 base pairs to be obtained in the PCR reaction. Place the samples in the thermocycler to perform the PCR. Finally, the thermocycler is programmed. For this reaction, an initial denaturation temperature of 94 degrees Celsius was used for 5 minutes. Subsequently, the 3 temperatures were programmed for the 30 cycles, with a denaturation temperature of 94 degrees Celsius for 30 seconds, an alignment temperature of 59 degrees Celsius for 30 seconds, and an amplification temperature of 72 degrees Celsius for 40 seconds. It is left at 72 degrees Celsius for 5 minutes to finish any partial amplification. And finally, a temperature of 4 degrees Celsius is programmed for an indefinite time to maintain the integrity of the products obtained.
When visualizing the gel of Figure 1, we can notice that in line 1 is a positive control, containing the expected amplification of around 450 base pairs and some remaining DNTPs. In line 2 is the negative control, which was water. This sample could have suffered contaminations since it presents non-specific products in the gel. Finally, the sample loaded in lane 3 was the genomic DNA sample, in which a very light band of the desired amplified product can be noticed. However, it also presents non-specific products with much greater clarity and leftover DNTPs. With Table 1, we can perform the stability analysis of the secondary structures formed by the primers. In this case, homodimers, heterodimers, and hairpins were analyzed. For hairpins, the tolerated range of delta G is greater than -3 calories per kg at least -1 per mole at least -1. In this case, for the two primers, the delta G values in the hairpins analysis are positive values, so its stability is good. For the self-dimmer and heterodimmer analysis, the tolerated delta G values are greater than -5 or greater than -6 calories per K at least -1 per mole at least -1, which led us to say that the secondary structures formed by these primers are stable.
To perform a successful PCR, some crucial aspects need to be taken care of. Not only specific concentrations and determined proportions for the master mix of the PCR is important, also it demands a sample DNA of great quality because it may contain reaction inhibitors that will affect the PCR to be performed efficiently or at all. Template DNA is often used from extraction procedures, and some of the components employed from these procedures may act as PCR inhibitors, as they are capable of reducing the magnesium ion, whereas the bivalent form of the magnesium ion is the one needed as a cofactor for the DNA polymerase. And some other components may interact with nitrogenous bases, and this will affect the polymerization. Thus, template DNA must be highly pure. In the length of the electrophoresis observed for this practice, there is neither evident protein contamination nor excessive salt content. Primers play another important role in PCR as they bind to their complementary sequence and elicit the polymerization of the target region. A primer is specific when containing 18 to 22 nucleotides and is easily denatured with each cycle. Because basic PCR contains two primers, the temperature at which both denature from their complementary sequence must not vary more than 5 degrees centigrade. So at the annealing stage, it is very likely for both primers to reach their complementary region in order to obtain as a result a new copy of the target region.
As they are single-stranded nucleic acids, they may bind forming hairpins and dimer structures. It is generally accepted that the calculated stability of a primer hairpin must not be lower than -3 kilocalories per mole. For dimeric structures, it is established that a value greater than -5 kilocalories per mole is ideal. Although 22 nucleotides is suggested as a limit size for a primer, 25 nucleotides is still adequate. By performing the stability calculations, we conclude that both requirements are met, and that hairpins and dimers form remains unlikely. Despite the primer heterodimer is the most likely to occur, its effectivity must be proven by the amplification of a positive control. As this positive control was correctly amplified, then the primers act properly in this PCR. The remaining reaction conditions prove adequate with this successful amplification. Otherwise, the reaction conditions would need some adjustments, including the effective elimination of reaction inhibitors. Negative control shows no amplification product at the size of 447 base pairs, as it is expected. Nevertheless, a band is observed at the size of 50 base pairs. This could be a cross-contamination of a non-specific PCR product from template sample in lane 3. The amplification on the template tested DNA sample shows very little amplification product, while a non-specific product is still more abundant. It is known from previous experiments that the concentration of contaminant PCR inhibitors is very high in this sample, which could cut the efficiency of the reaction and elicit the polymerization at non-specific sites. The contaminant concentration is thought to be the only difference between the positive control and the extracted sample, as the master mix was prepared in a separate tube and then subdivided. We may state that the target sequence is present in the sample, but the amplification barely occurred. A better extraction procedure needs to be performed, probably by a second phenolic extraction, or a more careful retrieval of the aqueous phase in which DNA is solubilized. Moreover, a correct quantification assay is important. Lastly, we must prevent false positives to arise due to sample loading.
In this practice, the critical points for a successful PCR were observed. In addition, the characteristics of the primers, such as temperature melting, temperature of alignment, the percentage of GC, and the length of the primers that were seen in the theoretical genetics engineering class were retaken. Regarding the results, the problem sample was not useful for the practice because it was not obtained amplified. It was concluded that the sample was not of a good quality or that the quantity was insufficient to perform the PCR. That sample were struck from the practice before. For the electrophoresis analysis, there were loaded 3 samples, the positive control, negative control, and the problem sample, but just the positive control were observed as a product of the PCR on the agarose gel.